MMI 133 extra review for the final exam. Here's a little Calvin and Hobbes for you. Ah, good, a true or false test. At last, some clarity. Every sentence is either pure, sweet truth or a vile, contemptible lie. One or the other, nothing in between. I'll just try flipping a coin. So let's talk about that final exam, shall we? Final exam is next Wednesday at 1 o'clock on Moodle. It's going to cover all of the topics from the entire semester, although the focus is going to be topics 11 to 14. Here's how I'm going to do it. There's going to be 40 questions of multiple choice, and what I'm going to do is that about 30% of those questions will be from topics 1 to 10. So really what I'm going to do is take 2 or 3 questions from every topic through the entire course, including topics 1 to 10. Your focus should really be on topics 11 to 14. So I would recommend maybe spending an hour or two on topics 1 to 10. Go through the notes. If there's any area that you're a little fuzzy or unclear, write down some questions, send me an email, but focus and do notes on topics 11 to 14. All these other sections, matching, fill in the blanks, short answer, they will all be containing, they'll all be directly from topics 11 to 14. Matching we've seen before, fill in the blanks we've seen before. I will probably address what some of those questions will look like in a future lecture or video, and short answers, these will be case studies. Overall, this will be 80 marks worth 40% of your final grade. Here's another cartoon of Roger cramming for his final exam in microbiology. So I wanted to show you this. This is a mind map made by a previous student of mine. I asked her if I could share it with future students, and I just encourage you to do something that involves taking notes. Mind maps are one way to do it. Here's another mind map. This one was done by kind of software. Here's some other notes. These are a little bit more organized. Make some notes. Notes are going to help your brain to remember things. Your brain will only remember maybe 5–10% of what I say in a video or a lecture. You start writing things down and you can double that pretty easily, even if you never go back to your notes. If you make good, careful, thorough, organized notes, it's only going to help you to know this stuff really, really well. So here are topics 11 to 14. I wanted to basically, for this video, just spend a little bit of time going over some study questions. So here's the first study question from topic 11. What 4 factors contribute to nosocomial infections? So what was a nosocomial infection? Do you remember? A nosocomial infection is an infection caught in a hospital or other healthcare setting. These are also known as hospital-acquired infections. So you may have seen this, HAI, as an acronym attached to something like HAI-MRSA, means hospital-acquired infection of methicillin-resistant Staphylococcus aureus. Or sometimes people just have HA, they don't put the I in there as well. So what are those 4 factors? This is basically right out of the notes. That's what the slide looks like. Here they are. First one is pathogens are already present in the hospital. That's right, that's a no-brainer. You have sick people arriving in the hospital bringing their germs. This was actually a huge contributor to this COVID-19 crisis in Italy. They have large hospitals, and sick people coming with the COVID-19 disease would be spreading the virus to all sorts of other patients in the hospitals, which led to a lot more infections. Notice my other note here that some of these may be drug resistant. That can also be a bad thing. Number 2, patients have compromised immune systems. Some of them are intensive care. Some of them may be old or elderly. Some of them are sick with things like AIDS or other things that are compromising their immune system, which makes them susceptible to infections already. Number 3, we have all sorts of people buzzing around in the hospital that can be vectors. Other patients, staff, visitors. You can imagine all of the things that are touching one patient and then maybe touching doorknobs and other things and then touching other patients. Lastly, hospitals are home to many invasive procedures such as surgery or catheters. Catheters, of course, are probably the #1 reason why people get hospital-acquired infections because they're so common. Here's another question. What are the 4 sources of pathogens? Sometimes people get a little confused about this question here. What does source mean? Really, in this case, I'm referring to this, a reservoir of infection. And again, this is directly from the notes. And there were 4 reservoirs that were discussed in topic 11. The first one is our normal flora. So just like the case of the catheter infection, that's usually taking an organism from the skin, introducing it to the urethra. There's all sorts of cases where Staphylococcus is introduced from the skin to various other parts of the body. Sometimes it's just deeper into the skin tissues from scratch or something like that. B, of course, many diseases are from other humans, so sexually transmitted infections fall into this category. All sorts of infections, of course, are caused by humans, and many do not actually have any other reservoirs. So measles, for example, is a good example of a virus that is not found in any animal and is only transmitted from human to human. Of course, if we can get disease from humans, we can also get diseases from animals. There's a big long list of diseases that fall into this category. Many people probably think of things like rabies, but there are a lot of diseases that people get all the time that are actually from animals. A good example is West Nile virus. The reservoir for West Nile virus is birds. So birds that include crows and robins and blue jays. And what happens is a mosquito will bite an infected bird, and then the mosquito bites a human. Very rarely is West Nile transmitted human to human. And lastly, we have abiotic sources. So we're talking about soil and water and things like that. Abiotic meaning non-living. A good example of this is botulism. Botulism is caused by Clostridium botulinum, which is a soil organism, and those endospores from that organism can get into food and then cause botulism. So here's a question from topic 12. It says here, a patient of E. coli dies of E. coli infection after an intestinal puncture. What is going on here is really the question. So this is really just a classic example of an opportunistic infection. So what is an opportunistic infection? This is where a pathogen can take advantage of a unique situation that's not normally available. And there are really different categories of opportunistic infections. First category is a person has a weakened immune system. So if you think about somebody with AIDS, they get all sorts of weird infections that most normal people do not get because their immune system is weakened. Rare cancers, rare fungal infections, pathogens that don't normally cause disease in healthy individuals. An altered microbiota, which means we have our normal flora is being changed in some manner. A good example of this are vaginal yeast infections, where some women might take antibiotics. The antibiotics kill the bacterial lactobacilli in the vagina, and this leads to an overgrowth of candida yeast, leading to a yeast infection. Third is a breached integumentary barrier, or really this means introducing one organism from a natural anatomical site to another anatomical site. I'll get back to that in a moment. Fourth category is what I call other complications. Sometimes there are other things that are going on with individuals that sort of don't really necessarily fit into those categories. And for example, I'm thinking of cystic fibrosis. So cystic fibrosis is a genetic disease, and people with cystic fibrosis, they have an extra thick mucus in their lungs, and this makes them susceptible to certain bacterial lung infections. So let's take a look at this one here, a breached barriers or introduction from one anatomical site to another. This is what has happened up here. E. coli infection is after an intestinal puncture. So E. coli normally lives in the intestine, and now it's ending up in the blood, and it's becoming a very serious infection that actually kills this particular patient. So that's really the answer to this question, is you have E. coli going from a place where it's not causing harm to human health to a place where it is causing harm. So here's a question from topic 13, comparing humoral and cell-mediated immunity. So I want to take a little bit of time kind of picking these two things apart because they're a major part of topic 13-2, which is of course adaptive immunity. So humoral immunity. Humoral is an old word that means blood. So that's where the name comes from that. But really we're talking about our B cells, and the B cells actually do a lot of their work in the blood and also in the lymph. So that answers the first part of this question. The primary type of immune cell are the B lymphocytes. What about cell-mediated immunity? That's the other one, so that would be our T cells or our T lymphocytes. B, of course, stands for, not blood, but bone marrow, and the bone marrow is where these cells mature. This is where they undergo clonal deletion. Remember what clonal deletion means? It means that our body is actually killing off any immune cells that may be self-reactive. The T cells, there's our T, they do this in the thymus. So that's where these cells get their names. So this says, what type of pathogen or foreign substance is recognized? So B cells secrete antibodies which really target smaller things. So what do I mean by smaller things? Things that are freely circulating in the blood or other tissues. So this includes things like viruses. Viruses are pretty small. Bacteria are pretty small, and other foreign cells, smaller cells. So maybe yeast might be the largest type of cell that can be attacked by the humoral immunity. We're also talking about large molecules, so protein toxins. So our vaccines against the pertussis toxin and diphtheria toxins are actually activating the humoral immunity. This may include some allergies, like allergies to penicillin can also fall in this category. Those are molecules that people can get in their blood and can have allergic reactions to. So what about the cell-mediated immunity? These are larger things. So of course we're talking about large parasites such as worms or other large pathogens. But we're also talking about our own cells that are infected by viruses. So when viruses infect cells, they'll often start producing viral glycoproteins that can be recognized by the immune system. And our own cells are too large to be destroyed by phagocytosis, so instead they are destroyed by the cell-mediated immunity. We'll talk about that on the next portion of this question. This also includes cancer cells or tumor cells. These cells can be recognized by the immune system, and actually our body clears them on a regular basis, which is a very good thing. So the next question, it says, what happens when cells are activated? Humoral immunity, as we talked about, involves the B cells, and the B cells secrete antibodies and make memory cells. How do antibodies kill the pathogen? Go back and take a look at your notes. There were 4 things that antibodies did to kill the pathogen. Most of them lead to opsonization, and by that we mean resulting in phagocytosis. How does the immune system remember the pathogen? Memory cells, of course, are made, which is why we have vaccines to try to stimulate the production of those memory cells. What about the cell-mediated immunity? These become cytotoxic. So cytotoxic, that is basically, I described it in lecture as the kiss of death. So how does this kill the pathogens? What they do is they attach onto the pathogen, and generally they do things like secrete toxins, and those toxins will destroy the host cell. These things are usually too large to be killed by phagocytosis, so they have to be killed by cytotoxic killing. How do they remember the pathogen? Again, memory cells. These wonderful memory cells help us to have long-lived immunity. So this brings us to the end of our first review video. I hope you enjoyed it. Please send me any questions that you might have, and I'll incorporate them into one of my next videos. Just to finish off, here's a little Calvin and Hobbes for you. Here he goes. Hocus pocus, abracadabra! I command my microbiology studying to be done. Be done! Flip, 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 flip. Rats. Anyway, I hope your studying goes well. Please send me lots of emails. I will do my best to get back to you in a timely manner. I hope you have a wonderful day and you're staying healthy.